We're thrilled to welcome Zach Sweeney, CEO and co-founder of Interline Therapeutics, to the show today. Zach, thank you once again for joining us.
Thanks for having me.
Let's kick things off. Zach, can you provide a brief personal intro for us today?
Sure. I'm trained as a medicinal chemist. I have worked in a number of large and small pharmaceutical companies. My training was largely in reaction kinetics and stereoselective catalytic transformations. I didn't, when I began my career, really understand how drug molecules were assembled. I learned that on the job, but I've really enjoyed working in drug discovery. My role has largely been to broadly help teams and companies develop the insights that they need to run really informative clinical studies. And I've been fortunate enough, together with my teams, to be able to identify a number of molecules that have moved into clinical development. A couple of experiences that are maybe relevant to what we'll talk about today were, going back to my transition from my undergraduate work to graduate work. As I mentioned, I ultimately trained in chemistry, but I was really attracted to biology because of the excitement at the time regarding the integration of genetic information with all of the new tools of molecular biology. And Linus Pauling was actually present at the undergraduate institution at the time. And, as I'm sure you know, he was really instrumental in uncovering for the first time the molecular basis of disease. So how a genetic variation results in a change in a protein and ultimately the manifestation of an endophenotype and disease in the context of genetic variants that lead to sickle cell disease. And so that was really fascinating to me, but really genetics was at a relatively nascent stage, at least as it was taught at the undergraduate level. I remember, really being interested in the broad global genetics of lactose intolerance. And that was like a major topic in one of the courses that I took, but there really wasn't much more information regarding, again, like the genetic basis of disease and the connections to proteins and mechanisms. And as a chemist, I was really interested in atomic level, what I call now like high resolution molecular mechanisms of disease. And again, just to frame it maybe for the audience, and this wasn't that long ago, there wasn't a good understanding of what common disease genetic risk data would look like. Would an average individual that suffered from a relatively common disease like Alzheimer's, would they have like one single mutation that strongly predisposed them to disease? And would all common diseases effectively be aggregates of lots of different somatic mutations or germline mutations that had a very strong effect size? Or would there be some different profile? And over the years, we've understood that common diseases aren't really like rare diseases. And rare diseases and early onset diseases, oftentimes you have one major genetic driver, but for most common diseases, there doesn't seem to be one major risk driver. Even that's true for many cancers. There's not just a single genetic variant that drives all of the cell signaling and cell proliferation. So we've learned a lot in that time, and I've been really interested in genetics from the very beginning. The reason that I mentioned rare diseases, I think, is in part because I had to decide whether to go to graduate school in chemistry or whether to pursue rare disease immunology research, actually, because at the time, again, the tools that were available to most research labs didn't really allow for the genetic study of common diseases. They allowed for more genetic study of familial diseases and rare forms of diseases that maybe had manifested in phenotypes that resembled common diseases. So the other key thing that was happening at the time was the HIV pandemic was really, profoundly affecting, I think, the lives globally, certainly the lives of people in the United States and in the Bay Area where I was. And ultimately, all these things kind of came together to encourage me to eventually transition from my postdoc to work at Roche in the Bay Area, focused on antiviral drug discovery. And that was really amazing. Going back to genetics, there was kind of an advantage almost if you were working in antiviral disease in viral diseases, because you could use viral genetics to inform the mechanisms that you were trying to interfere with as a medicinal chemist, and that was information that was easier to acquire. And there were really starting to be very interesting publications talking about how different changes in human genetics, particularly in proteins like CCR5, could protect from what was by then a common disease, HIV infection. So again, kind of a re-entry for me at that point, starting my industrial career and things that had really interested me for a long time and continue to interest me and fundamentally form the basis of Interline Therapeutics, which is the company that we'll talk about today.
And that's a perfect segue. And thank you so much for sharing more about not only your professional background, but the motivations at each step. I think having that context, understanding what was cutting edge and what was state of the art, and what were the societal factors shaping disease as you were making these decisions, and what these motivating factors were, is really powerful. So before we dive too deep into Interline, can you provide just a brief overview of your startup for us?
Yeah, so Interline is primarily a drug discovery company. I think, it's been amazing to be part of the pharmaceutical industry and the biotech industry over the years. And I want to take the opportunity, I think, to maybe emphasize to your audience again how much has changed in the 20 plus years that I've been in the industry, and pointing to HIV and hepatitis C as two areas of disease where we've just made tremendous progress in the state of medical care. So, what we want to do at Interline is in some ways recreate the successes that we've had in key disease areas like that at scale, using genetic information for other more common diseases. So we're a small molecule drug discovery company. We have a portfolio of 6 programs focused on oncology and inflammatory diseases. For each of these programs, we think a lot about what I mentioned initially, how genetic changes are associated with changes in the levels or the post-translational modifications or the interactions of the proteins that are associated with those genetic variants. And the key capabilities that we leverage for our programs are really our experimental proteomics capabilities and workflows, and really advanced computational chemistry, cloud-based workflows that we use to find our lead drugs. And what we try to do is create value in 2 ways. The first is trying to understand mechanisms of disease that are very proximally linked to genetic variants at the protein level, as I mentioned. And by understanding these mechanisms, it really provides you with key insights with respect to how you want to modulate cell signaling pathways and what very specifically at high resolution you want to achieve with your molecules. And then, of course, we try to find molecules that are safe and well tolerated and very clearly correct those signaling pathways.
Yeah, that's great. So we'll take this time then and perfect pivot into more of the story surrounding Interline. So in 2020, the company was founded, as you had mentioned, leveraging a lot of recent advancements in the quickly moving, molecular biology space, genomics, and you're focusing on protein communities. And so we're interested in you chatting a bit about how Interline Therapeutics has come about, and then we'll dive more into the company.
Yeah, so I'm a chemist. You can think of that, like, ultimately I try to, another way to think about what medicinal chemists do, they're like molecular engineers. They have a very clearly defined problem, and you try to synthesize molecules that affect a very well-defined problem. And what I found over my career is that oftentimes, especially when we're working on really important areas of unmet medical need, the problem wasn't necessarily a molecular engineering problem. The problem that we were facing was a fundamental uncertainty with respect to whether or not the mechanisms we were pursuing were the right mechanisms, whether or not we'd identified the patient populations that were really suffering from dysregulated signaling for the pathways that we were trying to interfere with, and lots of other things. So in 2020, I was transitioning from my previous two jobs. My first job was as a senior director of chemistry at Revolution Medicines, which is a biotechnology company locally. And then immediately prior to founding Interline Therapeutics, I was the chief scientific officer and head of discovery at Denali Therapeutics, which is a company that was focused on neurodegenerative diseases. And in both of those jobs, I had the opportunity to really decide what those companies worked on, what kind of programs we should prioritize. And what I realized is that, again, some of the programs that we worked on could very much be characterized as molecular engineering problems, where you had a very clear connection between a genetic variant that predisposed you to disease risk, the changes in the protein that were associated with that genetic variant, and that informed you very precisely about how you might want to modulate that protein and the signaling pathway that you thought it was implicated in. But there were lots of other challenge problems that we worked on where it was less clearly defined. And so what I thought about when founding Interline was, again, how are those engineering problems, what are the best drug discovery problems to work on, and what are the highest confidence targets that my teams had identified and other people had identified. And you can go all the way for common diseases, and you can go all the way back to systems like PCSK9, where there are very clear mutations in the target protein. You have a very clear understanding of how those mutations affect interactions with LDLR, and ultimately how that modulating PCSK9 might affect cholesterol levels and risk for cardiovascular disease, for example, at least in some populations. That's a classic example. More recently, there are examples like TYK2, where there's a very interesting common disease application for TYK2 inhibitors, but fundamentally the therapeutic hypothesis is very well defined. That was really a molecular engineering problem starting at about 2015, when he started to understand how the coding variants in TYK2 were affecting TYK2 levels and interactions with cellular receptors that were mediating inflammation. So I just wanted to be able to start a company that focused on problems like PCSK9, TYK2, and if we sat down and had more of a conversation, we could probably think of 10 or 12 other, maybe 20 very well-defined problems like that. And what I found in thinking about that was, ultimately what the challenge is, again, like a proteogenomic problem, to kind of introduce a rather complicated, but I think important vocabulary word, where you're connecting genetic information and genetic changes to changes at the protein level. And there were all these new technologies that allowed us to do that better than we had before. And we'll get to some of those later. And so ultimately, we could use those technologies. We could use computational chemistry, as I mentioned earlier, to, I think, more efficiently align our discovery activities and deliver valuable medicines for patients. A lot of what I was thinking about at the time was, how do you do drug discovery really efficiently? How do you avoid the expensive phase 2 failures? You have high confidence molecular mechanisms, you have biomarkers. And how do you do molecular engineering very efficiently? You have best-in-class computational chemistry. So in 2020, I had the opportunity to really think about those technologies, and I didn't do this alone, I should mention. So I really had the opportunity with the transition from Denali to work with a really great group at Foresite Labs that had an incubator, and they were kind of working on an aspect of this problem. And I had a really great relationship with some of the people at Arch Venture Partners, and they were interested in this idea from the beginning as well. and then the idea broadly evolved, of course. I mean, it continues to evolve to this day as I started to talk to some of the key people in this organization, investors, scientific advisors. We were really able to kind of refine the overall concept and identify key programs, and then continue to refine our workflows in the context of what was most useful for the drug discovery programs.
Great. So Zach, thank you for sharing some background on, Interline, how you got started, and some of the challenges that you were seeing in the industry that were ripe for an innovation. So one interesting distinction that you made is that from your training, you were very used to certain problems which were perhaps, quote, engineering problems or a molecular engineering problem, where you noticed there was a huge unmet need or perhaps white space in even the fundamental understanding of what that problem should be, to then subsequently address from an engineering perspective. And so I'm curious if we could double click on that and explore where protein communities come into the play, and then segue into how you are leveraging an understanding of these communities to, as you said, lift that veil.
Yeah, absolutely. So to your point, I think, and to kind of the implication of your question, I've seen different problems, some that could be characterized as molecular engineering problems, some that could be characterized as, fundamental biological mechanism problems, and both, and oftentimes in our industry, we're working on both types of problems. There can be an urgent unmet medical need and enough biological insight that we devote tremendous resources to trying to develop modulators of a given pathway, even without important biological insight. So to compare, for example, antiviral drug discovery, which I mentioned earlier, to drug discovery in neurodegenerative diseases is perhaps instructive. And let's compare, for example, the task that a medicinal chemist has who's working on a modulator of CCR5 with the task that a medicinal chemist might have working on a modulator of a putative neurodegenerative disease drug target, something that modulates APP, amyloid precursor protein, or something that modulates a significant genetic risk factor for something like ALS, say C9orf72. So in the first case, the CCR5 problem, you have human genetic variation that is protective for disease. I think that's really important, broadly protective across populations. There's, of course, certain strains of HIV that for which CCR5 inhibitors aren't efficacious, but for the most part, the major circulating virus, CCR5 inhibitors are efficacious. And how could we have predicted that they would be efficacious? Well, you have key human genetic variants, the Delta 32 variant in human CCR5, that was found to protect broadly people from infection against HIV. And the mechanism, the proteogenomic connection, was very clear actually from the beginning, even in the mid 90s. And that was that you had different ability of the human virus capsid, HIV capsid, to interact with the receptors that were required for cell entry, CCR5. So the capsid could interact with the wild type version of CCR5. It didn't interact as efficiently with the Delta 32 variant. And as a medicinal chemist, you knew that if you could just mimic the Delta 32 variant and block the interaction between the HIV capsid protein and CCR5, that you had a great chance of affecting HIV transmission and infection, or at least disease progression even. And there are, of course, biomarkers that could be used for clinical development and other things. But again, just focus on the proteogenomic basis and the fundamental insights that form the basis for the extensive drug discovery efforts in that area. And then if you compare that to the state of knowledge in neurodegenerative disease, it's really striking. I mean, when I started working in neurodegenerative diseases, and still, people tried to model neurodegenerative diseases by cutting neurons and seeing how neurons died. so very primitive insights. And to go back to some of the targets that I've mentioned, I'm not sure, even after all of the drug discovery efforts on amyloid that have ultimately gone into the clinic to try to modulate the progression of Alzheimer's disease, that most people that work in the industry have a very clear idea of the genetic rationale for focusing on amyloid. We don't really understand what amyloid precursor protein does. A lot of the patients that we characterize as being genetically predisposed to disease don't have genetic variants in amyloid precursor protein. They have genetic variants in another protease. So it's kind of a much more confusing landscape with respect to what specifically you're trying to accomplish. And again, to boil it down, the reason that it's confusing is because you don't have a clear connection between the genetic variants that are associated with the proteins, and ultimately how those genetic variants change the localization, the levels, or the interactions of the proteins of interest. C9orf72, just to close, is an example. It's a target that's the subject of drug discovery efforts very broadly in our industry. There are actually antisense oligonucleotides that are in development now. The idea is that the genetic variants produce a toxic protein, so that's the putative proteogenomic connection. But C9orf72 is actually a target or a protein that has probably a critical role in autophagy and other cellular processes. And as a medicinal chemist or a drug discovery technologist, I would argue that we're not really at the stage where we understand the basic target well enough to have the same level of confidence that we do for that target that we did for CCR5 or a lot of the other targets that led to very efficacious HIV treatments.
So thank you for providing some of that background. I think it really does help set the stage for exploring protein communities and being able to take more of a proteogenomic approach versus just a pure genomics approach, say, and to try to arrive at a new axis or new approach of saying, well, first, what should we target? What really even exists, and then pursuing that afterwards. So can you share maybe a little bit more on how Interline is applying these new insights and approaches to drug discovery?
Yeah. So I think what was changed, and to go back to the origin story, like why was the time right to found Interline? This is not a new idea. Proteogenomics, as I mentioned, goes back to Linus Pauling's publication in 1952. So it's a 70-year-old topic, and many people have pursued this concept. And so what has changed that allow us to try to understand not just rare disease proteogenomic connections, but common disease proteogenomic connections? And that's, I think, like a question of scale. So the first thing that really has happened recently is that we have new genetic data that we hadn't had before. So to go back very quickly through the eras that I've lived through, we had kind of the rare disease genetic era, and then we had the common disease genetic era. And the common disease genetic era was really defined by large-scale SNP studies where you were just, looking throughout the genome at kind of key points where a very few genetic variants, essentially, and lots of non-coding genetic variants. And what was discovered is that a lot of these genetic variants influence the risk for disease, but they only had a small effect size. And linking them very specifically to proteins was sometimes a challenge, or even when you could link them to proteins, it didn't really tell you, again, about all of the information that you needed, protein interactions, for example, to tell you how you might try to modulate that target to identify new therapies. So more recently, there's been the next version of the next genetic era, which is defined by really whole exome sequencing data. And so that, combined with much larger scale studies, has allowed us to identify rare but coding genetic variants that have a larger effect size, and importantly, change the protein in a critical way. Oftentimes they change the protein expression levels, but sometimes they don't change the protein expression levels and they change the ability of that protein to be ubiquitinated or phosphorylated, or can be associated with the change in the interactions of that protein. So lots of new genetic data for us to work with. That's the first big change. The other big change is the proteomic technologies have evolved so much in the last 10 or 15 years. It used to be that you could, with the standard instruments, identify maybe 1,000 or 2,000 proteins in a cell in a given assay, and now we can routinely measure at Interline 9,000, 10,000, 8 to 9,000 proteins really at scale. And you can do these kind of experiments very quickly in ways that you could. And so proteomic technologies, mass spec based technologies in particular, have evolved. Model availability in the era of CRISPR technologies has really changed. We can, instead of just overexpressing a protein or knocking down a protein using siRNA or CRISPR, we can knock in a tagged version of a protein or a variant of a protein into the endogenous locus. And you can imagine how important it is when you're trying to understand native protein interactions to not overexpress the protein and look for new interactions, or knock out the protein and completely remove it from the relevant community, but to study it at as close to endogenous levels as you can. And the ability to do that in relevant cell lines quickly has changed with the introduction of CRISPR and other genetic engineering approaches in just the last 5 years. And then finally, there's been a big change in the technologies that we use for molecular discovery. So most of my career has been spent identifying modulators of enzyme proteins. But if you actually take a less biased view of what is associated with disease, you'll identify lots of proteins that have no apparent enzymatic function. They have what's known as a scaffolding function. And so they bring proteins together to form these key signalosomes that mediate signaling transmission. And it's the formation of these signalosomes that can be dysregulated that drive common diseases in many cases. So the question is, how do you identify drugs that don't just modulate enzymes, but that modulate scaffolding proteins and presumably the interactions that they're trying to mediate? And so, most of the time we work with high throughput screens to identify the initial lead matter in drug discovery. And in the last 5 or 6 years, especially with the introduction of DNA encoded library or mass spec based affinity screening technologies, you can identify lead molecules that bind to your protein of interest more quickly. And so you can use that technology in conjunction with other protein interaction technologies, just standard FRET-based assays, for example, to, I think, more efficiently identify drugs that modulate protein interactions than you used to be able to.
So if I was to summarize in that case, you're doing a lot of work at the early stages with protein mapping of these communities, creating that large-scale atlas of information, linking it back to the genetics of common diseases primarily, and then using that novel information as the basis of your drug discovery platform. Did I summarize that properly?
Yeah, exactly. So I'm, I got caught a little bit in the technologies and what's changed, but the fundamental approach is, as you just articulated, to try in the best cellular models to understand exactly how the genetic variants that either protect from disease or predispose you to, or that increase disease risk manifest in one of 3 ways. How do they change protein levels? How do they change post-translational modifications of that protein? Or how do they change the protein interaction? So we try to understand the protein interaction network using mass spec-based technologies, using computational structural biology, and then we try to functionally validate the relevance of those interactions using standard functional genomic approaches. And with that as kind of foundational evidence for the molecular mechanisms, then we use the technologies and computational chemistry that I mentioned to identify drug leads and progress our drug discovery programs.
And as you look at those links and as you take this very protein focused approach, linking it back to the genomics and those mutational changes you're just describing, are you also looking at the phosphorylations, the ubiquitinations, and understanding maybe even the methylation at the genomics level? Or is it taking it one step down and then building on top of it as you gain this deeper set of knowledge?
Yeah, for the most part, I think we are not directly anticipating what effect the genetic changes have on some of the post-translational modifications that you mentioned, but we're trying to study the wild type version of the gene, the genetic variants, or the protein in different contexts, and then identifying those post-translational modifications. And the reason that they're important is, again, because they change the propensity of a protein variant to interact with other proteins. And then they also function as biomarkers of drug action. So we can use the changes in these post-translational modifications in the clinical development of our medicines by monitoring how they change with drug exposure in clinical studies and preclinical studies. So the proteomics platform is broadly useful for molecular mechanism identification and then also for biomarker discovery.
And anytime you can link a strong biomarker to a patient population, you greatly increase your chances of drugs positively impacting those patients. I would love to just wrap up here a little bit and ask, do you have any lead programs you're excited to talk about and maybe give a very brief overview on?
Yeah, we are still in the stage where we're not disclosing exactly what our lead programs are, but I can describe them generally for you. We are focused on, we have 3 waves of targets. You know, the first are kinase interaction modulators. It's been discovered that the enzymatic activity of kinases, so kinases are major drug discovery targets for the pharmaceutical industry, as probably most of your audience knows. What's I think been underappreciated historically that is more appreciated now is the key role that protein interactions play in the function of those kinases. So as I implied earlier, the first generation of molecules mostly just focused on the enzymatic activity of kinases. Kinases transfer a phosphate from one protein to another protein. And what was discovered, for example, in BRAF program, other kind of key oncology programs, was that it was oftentimes insufficient just to modulate the enzymatic activity of the protein, and you had to also modulate the interactions of that kinase. And so there are a couple of opportunities early in our portfolio that we've identified where we're pursuing those targets. We have also identified key genetically validated targets that have no enzymatic activity, scaffolding proteins. And these can modulate a number of different interactions. One's a modulator of a transcription factor, or binds to a transcription factor and seems to modulate its activity. So it's a transcription factor protein community drug discovery opportunity. And another is a signalosome that's involved in inflammation. Again, a scaffolding interaction where it's known that levels really drive disease risk and that you can modulate the inflammatory process just by modulating the levels of the interacting proteins. And then the third wave in our portfolio that we're really excited about are interactions between ligases and key proteins that drive either cell proliferation or inflammation. And so many companies are interested in molecular glues, for example. Now, molecular glues, by definition, stabilize interactions between proteins that facilitate the degradation of one of the proteins and the target protein. There's a native weak interaction, and the molecular glue essentially stabilizes or facilitates that native interaction. So in order to discover molecular glues systematically, I think what you really need to do is understand as best you can the native interactions between your target of interest and the machinery that is facilitating the degradation of that target. And then you can very rationally, hopefully, identify activators of that interaction. So the third wave of our portfolio is really interaction activators. And so we developed a number of assays and are pursuing those drug discovery opportunities as well. So hopefully you can tell I'm really excited about programs and all of those three waves, and look forward to in the future revealing specifically what those targets are.
And even the teasers get us excited. And I know just listening, the fact that you've been able to develop such a comprehensive, almost tech bio platform here, and use that to design 3 waves of therapeutics targeting different aspects of disease biology is incredibly interesting and a milestone to be highlighted for sure. So I know we're going to be excited to hear when you're able to share in a little bit more detail and to drill down a little bit deeper into those accomplishments. You've been leading Interline now from its inception to over 90 million in funding from phenomenal investors, as you mentioned earlier, such as Arch. And we'd just love to learn a little bit more. What have been some of the greatest challenges in the journey along the way?
I think that the major challenge when you're growing a company is trying to bring together all the parts that will synergize with one another before they're actually present as individual entities. So how do you build a team without a portfolio? How do you build a technology without a team? How do you attract funding without a portfolio and a team and a technology, right? So you have to really, I think, be very strategic and accept your kind of iterative limitations and stay true to your long-term vision. So, I think the challenge has changed over time. And, initially the challenge was identifying an area of science that I thought could be transformative, that could change the efficiency with which we ultimately identified drugs successfully. And then I had to learn how to kind of articulate that vision and get people excited without a team, without any technology experts, and without a portfolio, And so then I think the next key thing at Advantage was to identify real drug discovery targets that, without any of the novel insights that I just mentioned, could provide Interline with the opportunity to demonstrate and grow its technology capabilities, but also very quickly get started on real drug discovery programs. So the second part was, after the idea was, to try to identify 4, 5, 6 interesting drug discovery programs that were consistent with the philosophy that I just mentioned, where we could fill in key gaps, but where there was pre-existing information that allowed us to get started, right? And we want, I think it's important for any biotechnology company in particular to have a key kind of path forward for first-in-class or best-in-class molecules, like in a real way. But you have to be able to start with pre-existing information at the same time. So that's a relatively narrow intersection. And then with those things together, the task was really then to build the team. And, that has been amazing. We're 50 people here today. I think has been the most amazing accomplishment, is to come back like I have from vacation in the last couple of weeks and meet 4 or 5, amazing new coworkers. So building the team was the next challenge. And, you just have to understand that, and then the team, I'm including founding scientific advisors, key collaborators, contract research organizations, but also, of course, Interline full-time employees. And, the first 3 are harder than the 3 after that. And, once you build a team, then everybody's recruiting to kind of build your technical capacity. And I think that the way that, again, you attract that team is by having a very clear vision and something for them to really get started on. So that's why I kind of mentioned all of those components have to come together. And then with those 3 components, I think the challenge has been just to demonstrate the importance of the kind of data that we think is going to be, again, transformative and be the kind of data that people are going to want to focus on in the future for drug discovery. And we've spent the last year doing that. And we're really excited about the data that we've generated for all of our portfolio programs. And now we're really starting to see the synergies between all of the components that I just mentioned.
Well, thank you for sharing a bit about the history of Interline, some of the challenges you've faced, certainly the successes and a lot of the exciting opportunity coming forward in the future. So we have a couple quick rapid fire questions just to cap things off. So taking that next step in our sequence, what's coming next for Interline?
So there are some, key technologies that I think, I alluded to maybe earlier, but that we really need to continue to integrate. So I'll tell you about platform growth first. So we've initiated collaborations with certain key computational structural biologists. And this is going to be a really important area for us to master, because with the advent of AlphaFold, which allows you to use machine learning and all of the structural data to anticipate the structure of a protein, a single protein, from the sequence of that protein. The next challenge is going to be to anticipate not just the structure of that protein, but all of the different interactions that protein engages with, right? So this is going to be the next frontier, I think, for computational structural biology. And without recounting everything that I've said in the last 45 minutes, you can see how important that is for us. So we're going to continue to grow that group. We are very interested in our ability to monitor protein levels and protein interactions quickly in different cellular environments. And so how best do you integrate that with functional genomic screens? So CRISPR-I screens, CRISPR-A screens, that'll be a key area of growth for us. And then the third thing that I think I'm really excited about with respect to our platform is the computational chemistry component, which I haven't emphasized in this conversation. But there are, I think, really rapid, interesting advances being made in our ability to do short molecular dynamic simulations. To identify different protein conformations, understand how that impacts interactions, but also understand how that impacts the binding of molecules. Companies like Relay and many others have been working on this intensively for the last 7 years, but I think we're continuing to see improvements there. And so short molecular dynamic simulations coupled with what are known as relative binding free energy calculations, I think should really come together to allow us to do drug discovery, the molecular engineering that I mentioned, more efficiently. So those are the platform changes, and you'll see over the next, I think, 1.5 to 2 years, the implementation of those, the continued implementation of those technologies and insights and the application to our portfolio. From a portfolio perspective, we're heavily invested in our programs. So you'll see novel molecular mechanisms, you'll see molecules that clearly correct protein interactions that are moving towards the clinic from Interline. And I hope in 2024 we hope to be in the clinic with a couple of programs and be much further along with a couple of other programs. I mean, ultimately, when you're talking about a goal as audacious as trying to improve the efficiency of drug discovery, you have to demonstrate that you can efficiently discover drugs. And so hopefully we'll be able to demonstrate that in the coming months with the Interline portfolio.
Well, hopefully, and that's such an exciting project to be working on, in the industry right now. So as we've capped things off, we just want to ask if there's any other closing questions that you might have, any shameless plugs or last minute thoughts.
Not really shameless plugs. I think ultimately what we're all trying to do is help people. And then the sub goal to that is, how do you create value for society? And I think in a capitalist, in any economy really, but certainly in our economy, you create value by trying to do things more efficiently. It's not that we can't discover drugs now. We have a 4% success rate or whatever it is. But I hope that the question that people that have listened to this point in this podcast ask themselves is, how can we do this more efficiently, more systematically? And I've proposed one approach where you very much focus on causal genetic variants and try to understand the proximal phenotypes at the protein level and then work forward in that way. And I'm sure there are many other people, and focus on small molecules because they can efficiently be distributed to all sorts of countries and people take, but there are other exciting opportunities out there, but I really think that they should be framed in two ways. One is, do they deliver value and new efficiencies for our society as a whole? And two, are they as rigorous as they have the potential to be, given all of the technology advances that we've made in the last 20 years? So that's, I guess, my closing hope for the audience is that they can improve even on what we're doing here and find new ways to bring value to society and the patients that we hope to serve.
Well, and that certainly is the goal of the industry as a whole. So I think that's a great way to wrap things off. Thank you again, Zach, so much for an amazing episode. We're very grateful for your time. Thank you.
No, thank you, Michael, and thank you, Chris. It's really exciting to meet you. And next time you're out in San Francisco, you have to stop by the Interline offices. Open invitation.
Thank you, sir.